Dear friends, welcome to the lecture number 5, Zoo 301 Physiology. Up to now we have seen how the action potential is being taken place, how the nerve impulse passes from axon, then from axon hillock, how it originates and passes to the nerve axon. In between, how depolarization and repolarization takes place. After depolarization means movement of sodium ions, how the membrane goes to reset position. As soon as the aggregation of potassium ion increases, how repolarization starts. In between that, how hyperpolarization occurs, that we have already studied. Now in this today's lecture we will see how the current flow takes place around the impulse which passes through the axon. In the axon, the impulse or the signals passes slowly one by one, and how the impulse is being carried out or passed out or facilitated, or we can say how the action potential works, that we will see in today's lecture. So current flow around an impulse of axon is picturized here. In last topic, these two simple diagrams of depolarization and repolarization is clear, because from this picture you will clearly understand how the current flow occurs around this impulse. So this is the axon. This is complete axon. In axon, this part is impulse which is passing continuously. During that passage of impulse, these positive ions and these negative ions, these positive ions and these negative ions, in between that, these negative ions which are moving again, how around this impulse, that is clear from here. You see, as soon as the sodium enters, the negatively charged outside and positively charged inside. As soon as the potassium moves outside, negative charge inside and positive charge outside. So this same mechanism takes place continually around the membrane. We have seen the resting membrane potential and working membrane potential, how it changes time to time as the action is taken place. And action potential, when it carries whenever neurotransmitters are secreted. Neurotransmitters are secreted when only as soon as the signal is to be received or impulse is to be received or information is to be conveyed from the message. And so these are the repolarization and depolarization, which maintains the balance of positive and negative ions continually around this membrane, and this helps to pass the impulse throughout the axon. So this is the current sink from which the impulse is passing. This is the extracellular fluid, and this is the intracellular material, and again this is the extracellular fluid. This whole complete is the axon, and this axon shows the positively charged ions ever outside, and as soon as they enters, it becomes negatively charged. As they outside, it becomes positively charged. So this is the current flow, or this is the current of ions flow, and this is the backflow of ions around this impulse for the maintenance of membrane potential throughout this axon. And therefore this diagram represents the current flow around the impulse of axon in unmyelinated nerve fiber, because this is a single straight fiber, there is no myelin sheath, and how the nerve fiber representing the movement of this positively charged ions, and the impulse is clearly indicates the straight flow of the impulse, and this arrow indicates the direction of propagation. And in this way the current flow around the impulse of axon is carried out. Second, propagation in myelinated nerve fiber. In last time we have seen non-myelinated fiber. Now we will see in myelinated fiber. The propagation and passage of nerve through myelinated fiber is generally called as salutatory conduction. Is generally called as salutatory conduction of impulse or message. So in last figure you have seen a single straight axon, and here in myelin sheath, this is the myelin sheath. This is continuous myelin sheath, and these are the nodes or gap. These are the nodes or gap, and throughout this the impulse is passing. Means whenever the impulse is passing, these ion gated channels at the node site works to activate and deactivate, repolarization and depolarization facilitate through these nodes only, and therefore this positively charged as they leaves and enters becomes negatively charged. As soon as they enter inside, it becomes again positively charged, and as they ready for move to outside, it again becomes negatively charged. And in this way, this alternate movement of ions to be hyperpolarized or take part in depolarized and repolarized is propagated in myelin sheath in this way. Means impulse which passes through this node is continually passes. This myelin sheath also plays a role to not allow these ions, but they are only passes through this membrane. And this diagrammatic representation is nothing but the local flow of current around the impulse of the axon. Now, this can be described in this way that the myelinated nerve fiber conduction depends upon the similar pattern of circular current flow as we have already seen in non-myelinated sheath. In myelin sheath is an effective insulator. This is the insulator. Therefore, this insulation is not allowed to enter directly, but ions cannot pass along the myelin sheath. You see, this myelin sheath is not allowing to enter the ions. Therefore, they passes through these nodes. But nodes of Ranvier permits the ions, and the action of potential is being carried out there too. And for this reason, the impulse is transmitted from nodes to node, and rather than continuously along the entire of the nerve fiber. The depolarization in myelinated action jumps from one node of Ranvier to the next. This is the jumping from one node to the another node, and this facilitates the action potential, or we can call it as a leaping, jumping or leaping of depolarization. Actually, the depolarization is the propagation here in the salutatory conduction of the nerve impulse to the axon. Now, rate of conduction of nerve impulse is different in different cells, or different in different types, or different in different type of nerve fibers, either myelinated or non-myelinated fiber, because the speed of conduction of impulse in these two different fibers is different, not same. And the basic principle of origin and propagation of nerve fiber or nerve impulse is the same, but both in non-modulated and myelinated nerve fiber, but the saltatory mechanism of conduction in myelinated nerve fiber increases the velocity of conduction more than 500 times. Means myelinated nerve shows conduction in myelinated fiber increases, and the velocity shows maximum up to high speed, that is more than 500 times or more than 499 times than the normal. And this rate of conduction of nerve impulse facilitates the action potential or passage of the signal. The rate of conduction of nerve impulse increases with an increase in the cross-sectional diameter of the neuron and with increasing thickness of the myelin sheath. As previously we have seen that the thickness of myelin sheath depends and causes the actual movement of the impulse or the signals, and the rate of transmission of a given neuron is constant. We will see in the next table, or we can clarify the idea of the rate of nerve impulse conduction through different nerve in different animals is shown here. The rate of conduction of impulse in vertebrates and invertebrates is different. Even though the nerves are everywhere same, but the structure, fiber and nature is different, and therefore the rate of conduction, the velocity and speed of conduction is different. So in nerve net in slanted animals, especially in sea animals of the species Metridium, the velocity in meters per second is 0.121 to 0.146, whereas the nerve cord in Annelida, especially in Lumbricus earthworm, is 0.6 meters per second is the velocity. In nerve fiber of mollusk, especially in the snail helix species, helix genus, is 0.4. In vertebrate animals, among frog, especially in non-myelinated fiber, the velocity ranges between 0.4 to 0.5 meters per second. And the giant fibers which are found in mollusca like loligo, or we can say sepia animal, is about 718 micrometer long, and its velocity is 27.3 meters per second. So it shows a very high velocity. In vertebrates, emeritus fish have 22 to 43 micrometer long nerve fiber, and its velocity is too high, means up to 50 to 60 meters per second, and therefore fishes shows a great activity in their physiology. Now saltatory conduction we have already studied in previous discussion. So what is the saltatory conduction, how it works in action potential in both the fibers, especially in myelinated fibers, and how its velocity in conduction of the impulse it changes, that we will see here in this paragraph. The saltatory conduction, from the Latin word saltator, the saltatory conduction is originated, and the saltator means to hop or leap, or we can say simply jump. How it jumps? Why it jumps? Because in myelinated sheath, the myelin sheath is not allowing the molecules or ions to enter there too, and therefore they directly jump to the another membrane where node is there and where myelin sheath is absent. Therefore the saltatory conduction or salutatory conduction is the propagation of action potentials along myelinated axons from one node of Ranvier to the another node of Ranvier, and increasing the conduction velocity of action potential. Instead of passing through the membrane, it jumps directly to the another node, and therefore the speed increases, and therefore the action potential shows great velocity of conduction of the impulse. In another way, the uninsulated nodes of Ranvier are the only places through which the axon ions are exchanged through the membrane or across the membrane, and the action potential is regenerated. As the action potential between regions of axon that are insulated by myelin sheath, unlike electrical conduction in a simple circuit, the jumping or leaping, or we can say saltatory conduction, takes place or occurs there in the myelinated nerve fibers. Now the next topic of our syllabus is the ultrastructure of skeletal muscles. In our first chapter of physiology, or in our first unit of physiology, the nerve structure of nerve and muscle fiber is there. So we have studied all about nerve fibers or nerve cells, nerve types, and its a detailed structure. Now we will see what are the skeletal muscles and what is the ultrastructure of skeletal muscles. As the nerves are specialized tissues or cells, skeletal muscles are also a specialized type of muscles among the different types of muscles. The basic type of muscles are skeletal muscles, smooth muscles, and cardiac muscles, or we can say voluntary muscles, involuntary muscles, and cardiac muscles. These are the three different basic types of muscles. So muscle, we can also call these muscles as striped muscles or striated muscles. So these skeletal muscles are generally attached to the skeleton or bones, therefore they are called as skeletal muscles. And as the name is skeletal muscles, they help in the movement of the skeleton of the body, and ultimately the whole animal moves there. The muscles which are found in association with skeleton is also known as skeletal muscles or striated muscles, because these muscles have different bands or striations on its whole length, and because of these fibers they shows alternate dark and light bands on the muscle fibers or muscles if we observe these muscles under light microscope. The muscles are usually in a partial contracted state, which gives muscle tone and make ready for the contraction under the stimulus preceding a complete contraction of that special muscle cells. The basic structure of muscle is a binded form of various micromuscles is called as muscle fiber. If you will see the microscopic structure of this muscle, if you cut the transverse section of these muscles, and if you see that there are various bundles of muscle fibers, and if you stretch one muscle fiber, then if you cut it and observe the internal structure, you will find again some microfilaments. Means this is the muscle fiber, these are the myofibrils, and in myofibril you will also see the microfilaments. These filaments make together the fibers, fiber make together the muscle, and muscle together form the muscular system. And this is the basic way of muscle structure. So these are the striations, or we can say bands, found on the muscle fibers. And the muscles are also a cell, a type of tissue. Therefore this muscle cell have this large nucleus which controls the various activities of these fibers and its activities. And therefore these are the large nucleus there. These muscles are composed of these myofibrils, and these myofibrils are composed of these myofilaments, or we can say microfilaments of the muscles. These are of two different types, that is actin and myosin. Therefore the myofilaments are of two different types, and the general structure of muscle fiber is either brownish or reddish. Now, the gross structure of muscle, if we study in detail, then we will find that it is totally composed of different fibers. And we have seen in previous figure, each fiber is made up of filaments. And these filaments are of two different protein molecules, that is actin and myosin. But some of these muscles shows a layer of or a coating of some protein materials. So they work differently, and that membrane material have a special name, lemma, means layer or coating. Muscle is composed of muscle cells, which are called as muscle fibers, because they are so long, they are cylindrical in shape, they are multinucleated cell and mostly resemble to the fiber than cell. Actually these are the cells, but as they are elongated and in the form of filament or fiber, they are looking like the thread or fiber, and they are arranged parallel. All these fibers are parallel to each other. Each fiber is multinucleated and the nucleus are located near the surface of each fiber. The bundles of these fibers are surrounded by collagen fibers and connective tissues to bind all these fibers together and to form a muscle to work properly for its facilitation of the physiological action. Each muscle fiber is enclosed by a plasma membrane. Enclosed by a plasma membrane called as sarcolemma. I told you previously that lemma is nothing but the envelope or membrane structure, which or covering structure. Therefore, the muscle fiber in Latin word is known as sarco, and therefore its membrane is lemma or covering, and therefore the muscle fiber enclosed in the membrane is called as sarcolemma. Furthermore, we have studied this structure of muscle, structure of their fibers, and arrangement or connection by membrane of collagen and connective tissues, and it is enveloped by the sarcolemma. Now, this fiber-like muscle cell is a special cell. We have seen the nucleus, and it also have a cytoplasm known as sarcoplasm, because the cell muscle fiber is known as sarco or in Latin word, and therefore its plasm, cytoplasm, is nothing but the sarcoplasm, which contains large number of mitochondria. Skeletal muscles or striped muscles, or we can say striped muscles, are the active muscles which facilitate the strength, or we can call the working of the strength of the body, and therefore they contain large mitochondria because they need a large amount of energy, as these mitochondria are the powerhouse of the cell, and they give the power, they give the energy to the nerve fiber to facilitate thereto with the associated organ. As we have studied, or as we are knowing here about the skeletal muscles, these muscles are working in association with skeleton, and we all are well known about the skeletal muscles, that these muscles are the strengthening or strengthened muscles of the body, and which lift the whole strength of the body. So this is the brief structure about the muscle and muscle fibers. Here we stop our fifth lecture, and in the next period we continue to study the ultrastructure of muscle, and here the fifth lecture is completed. So see you in the next sixth lecture. See you.